Hey, good morning, everybody. It's Tuesday, April 7th. I'm posting a new video today to give you some updates. I had a staff meeting yesterday. I've been also been getting questions by email from students and parents about things happening with your school. They probably haven't decided on a lot of these things yet, but we talked about it in the staff meeting yesterday. So yesterday we talked about what's going to happen the rest of the year, since you will be not coming back to school this year. You'll be continuing to do distance learning the best you can, going online, signing in daily, but it might be getting a little bit easier for some of you, or all of you maybe, because they're going to set up some type of schedule, most likely, in which the teachers will have a particular time in which, or day and time in which they're expected to give you assignments, or when they're due, when you're supposed to maybe sign online, get into Zoom if you do Zoom meetings with your other teachers. So you're not asked to be online with two teachers at the same time. It should be a little bit easier for you. The initial thing they reported to us, which I'm not saying this is exactly what's going to happen, but they might be doing like 2 classes a day. So that might be a little bit easier for you, because the past few weeks you might have been getting assignments from all your teachers every day, possibly, which might have been really tough, especially if you're at home helping with brothers and sisters and things like that, or you have other responsibilities at home to do. So that might make it easier for you. Just make sure you're using your time responsibly and still getting the work done. You will still be getting a grade. And that's another question too. Students have been asking me, will you be repeating 7th grade? No. As of right now, that's not been discussed. That should not be happening. You should just be still actively working, doing any assignments your teachers post for you to do, so you're better prepared for next year and you get the information you need this year. So if you have any other questions or you're not sure about other things, let me know. None of you are 8th graders, but if you have 8th grade brothers or sisters, they were talking also about promotion and what they're going to do with that. So they'll send out information about that. Yearbooks, they're going to set something up to order yearbooks online. And you'll be picking them up. If you already ordered one or you're going to order one, you'll have to pick it up probably sometime in summer or early next year when we go back to school, hopefully. And that was pretty much the meeting. The rest of the meeting was just teachers' information of what we should be doing. So I'm going to pretty much be doing what I've been doing. I'm going to do videos. I'm going to give you work to do, assignments that you need to turn in on certain days. If you have any questions or need help, remember, let me know. You can use Google Hangouts. I've been using Google Hangouts here and sending messages. I've been saying when I've been online, if you see a message like this, that means I'm online ready to answer questions and giving you updates. You can message me right back on here and let me know if you have any questions. That's pretty much it for updates. I'm going to go ahead and get into your reading for today. And some review, and like I said, tomorrow I'll be going through a video review. I'll plan another Zoom meeting probably for Thursday before you start your spring break, so I'll post that up Wednesday, tomorrow. Okay, so I believe I went over some of this with you already. I'm just going to review a little bit here. I talked about the genetic code and the order of the bases. You should have notes on this. This is from page 176 here that you should have already saw video on and done some reading and note taking. I also asked some of these questions on the Kahoot game. So let me scroll down and review this. So we talked about making proteins and protein synthesis. So the cell uses information on a gene on a chromosome to produce a specific protein. Protein synthesis takes place on the ribosomes. That should be a word that sounds familiar to you from way back beginning of the year, in the cytoplasm of a cell. As you know, the cytoplasm is outside the nucleus. The chromosomes, however, are found inside the nucleus. How then does the information needed to produce proteins get out of the nucleus and into the cytoplasm? So hopefully you're visualizing this and you remember the nucleus from inside the cell. And it's going to be asking, how does that information get in from the cytoplasm as it shows here with the cell. So we're gonna talk about this. I'm gonna show it in the video tomorrow. There we go. So the role of RNA before protein synthesis can take place. A messenger must first carry the genetic code from DNA inside the nucleus into the cytoplasm. This genetic messenger is called ribonucleic acid, or RNA. So that's ribonucleic acid. Remember I told you earlier in the year DNA is deoxyribonucleic acid. Okay, so this is a strand over here it's showing, and it's going to have this RNA. It's going to transfer this. It's going to go from the nucleus into the cytoplasm. Okay, so it says here, although RNA is similar to DNA, the two molecules differ in some important ways. Unlike DNA, which has two strands, RNA only has one strand. You're going to hear me comparing it to like a zipper, like unzipping a zipper like on a jacket. It's going to go from two strands to one strand. It's going to make a copy. RNA also contains a different sugar molecule from the sugar found in DNA. Another difference between DNA and RNA is in their nitrogen bases. So we went over that a while ago. So like DNA, RNA contains adenine, guanine, and cytosine. So RNA also contains those. However, instead of thymine, RNA contains uracil. So it's similar to DNA, but slightly different. Okay, so instead of thymine, it contains uracil. Okay, so types of RNA. There are several types of RNA involved in protein synthesis. Messenger RNA copies the coded message from the DNA in the nucleus and carries the message to the ribosome in the cytoplasm. It's basically making a copy and taking it somewhere. Just like if you were to make a copy of a math worksheet and give it to somebody, it would be like that messenger RNA. It's carrying it. Or like somebody that is producing something and shipping them out, whether it be letters in the mail and things like that, it's copying those and carrying it out. So another type of RNA called transfer RNA carries amino acids to the ribosome and adds them to the growing protein. So lots of things working here. And like I said, this will probably make more sense tomorrow after we do the video review. So this is the DNA molecule. Remember I talked about it like it's kind of like a twisted ladder. This shows it more here like in a small section where it looks just like a straight ladder. And it has these nitrogen bases pairing together. What's going to happen with this protein synthesis is it's going to like unzip. Like if it's a jacket that zips up, it'd be like unzipping. So this side would separate from this side. So let's continue on the next part here. So for translating the code, the process of protein synthesis is shown in figure 17. So it's going to be down here, and I'll review it with you. The first step is for DNA molecule to unzip between its base pairs. Then one of the strands of DNA directs the production of a strand of a messenger RNA. To form the RNA strand, RNA bases pair up with the DNA bases. The process is similar to the process in which DNA replicates. So we did talk about DNA replication a while back. If you want to go back and review or Google that or something, that's fine. But for DNA replication, we talked about it, how it unzips as well, and it finds its new pair. So cytosine always pairs with guanine. However, uracil, not thymine, pairs with adenine. So when it makes this copy, that's what's happening. It shows it here in step number 1. Okay. So it's going to say in the nucleus, the DNA molecule serves as a pattern for making messenger RNA. The DNA molecule unzips between base pairs. RNA bases match up along the DNA strands. The genetic information in the DNA is transferred to the messenger RNA strand. So you see it happening here. Here's the nucleus. The DNA is unzipping. This is going to be the messenger RNA. Okay? And then we're going to go into step 2 here. So step 2 is messenger RNA attaches to a ribosome. Okay? So it's going to have this messenger RNA here. It's going to have the ribosome here. And it's going to have this transfer RNA and amino acid. So it's going to be kind of finding like a new pair. It's going to have this copy here. And new ones are going to match up and align. So that's going to be step 2. Pretty much what it says there. That it's going to attach to the ribosome. Okay? Moving on to step 3. So step 3 is going to be molecules of transfer RNA attached to the messenger RNA. The bases on the transfer RNA read the message by pairing up 3-letter codes to bases on the messenger RNA. For example, you can see that a molecule of transfer RNA with the bases AAG pair with the bases UUC on the messenger RNA. The molecules of transfer RNA carry specific amino acids. The amino acids link in a chain. The order of the amino acids in the chain is determined by the order of the 3-letter codes on the messenger RNA. So it should be showing us step 3 here. So it's going to have this growing protein. It's going to have these RNA molecules. They're going to be read and the messenger RNA by matching up with these 3-letter codes. So they're going to be matching up, okay, forming these pairs that should be going along with one another. And then moving on to step 4, the protein chain continues to grow until the ribosome reaches a 3-letter code that acts as a stop sign. The ribosome then releases the completed protein. So it's going to have like a stop sign. It's going to automatically know at the end of these 3 pairs together, it's going to be a stop sign. That's going to be a completed protein. And then it's going to have a new one join by this transfer RNA. Okay, so that's what I wanted to share with you. We'll talk about mutations probably after spring break. Because tomorrow, this might have been a little bit confusing to you. Tomorrow I'm going to show a video similar to the meiosis one, and I'll be going through and giving my commentation during that video and giving you information that will hopefully help you. There's nothing again due today. I'll have something for you due on Thursday for you to complete. And if you have any questions, let me know.